Thank you, Professor. Good morning, ladies and gentlemen. As you can see, my topic today will be the diagnostic relevance of microRNAs in pancreatic neuroendocrine tumors. If you don't know what microRNAs are, don't be afraid. We will get to that point, and probably this meme will help you understand how they work. But first, I would like to tell you about the pancreatic neuroendocrine tumors, or PNETs as I will refer to them, but be careful, it's not peanuts, it's PNETs. Pancreas has exocrine and endocrine cells. Exocrine cells secrete digestive enzymes for the gastrointestinal tract, and endocrine cells produce hormones for the bloodstream. Neuroendocrine cells are endocrine cells mediated by the nervous system, and PNETs are the tumors of these cells. They can be hormone-producing or non-hormone-producing tumors. GRADE is a histological classification of these tumors, and based on their mitotic index, Ki67 index, and differentiation, we can sort them into 3 grades, as the table shows. The grading is done by the histological examination of biopsy samples, and now I will tell you about these indexes. The mitotic index is the number of mitotic figures per 10 fields of view using 400 times magnification lens on the microscope. And however, you can only see these mitotic figures if the cells are in the M phase of the cell cycle. And this picture shows how mitotic figures look like with hematoxylinosin staining. Ki67 is a protein that is produced by cells that are in the active phases of the cell cycle. So this is a perfect marker for cells that are preparing to divide. The only problem is that we cannot see this with basic hematoxylinosin staining. This is where immunohistochemistry comes in handy. In a nutshell, with anti-Ki67 antibodies, we mark these cells, and they will be brown under the microscope. And you can see that in a grade 3 tumor, there are much more dividing cells, or cells that are preparing to divide. When pathologists decide whether the tumor is well or poorly differentiated, they observe whether the tumor, or how closely the tumor represents the original tissue. And I will show you an everyday example for this. These corn cobs depict this perfectly. On the right-hand side, you can see that the cells are in perfect order, but there are some that has a different morphology, like in a grade 1 tumor. On the left-hand side, the cells are a little bit disordered, but you can still see the original structure, like in a grade 2 tumor. However, on the southern cob, the cells are in complete disorder, and you can't even see the original structure like in a grade 3 tumor. And finally, you can see the 3 grades beside each other, and on the Ki67 stained sections, you can see that as the grade is increasing, more and more cells are preparing to divide. Now back to microRNAs. They are short, stable, non-coding RNAs that play a role in the post-transcriptional regulation of gene expression. They attach to the mRNA and prevent them from binding to the ribosome or promote their degradation. And this is what this meme is trying to show you, that microRNA stops the mRNA from binding to the ribosome, hence inhibits the translation. Their expression is tissue and tumor specific and may be characteristic of the grade. The cells are actively secreting their microRNAs into their environment. Thus, we can detect them from archived tissue blocks and body fluids as well. They are heavily researched due to their possible therapeutic, prognostic, and diagnostic relevance. In our research, we are searching for microRNAs that could show a significant difference in expression according to grade, and thus could be suitable for grade determination. In the long run, we plan to measure the circulating counterparts of such microRNAs to find minimally invasive biomarkers that could complement or replace the current histological method. The histological examination is time-consuming, invasive, as it requires biopsy sampling, and it requires an experienced pathologist. The measurement of microRNA expression, however, is faster, less invasive, as it can be done from blood samples and can be done by a lab assistant. Based on literature data, we have chosen 4 microRNAs that has been described to show a significant difference according to grade. And these are miR96-5P, 106B, 130B3P, and 194-5P. We've collected already diagnosed tissue blocks from the 2 departments of pathologies at Semmelweis University and isolated total RNA from them. From these samples, 16 were grade 1, 18 were grade 2, and only 2 were grade 3, so we could only compare the grade 1 and the grade 2 tumors to each other. With reverse transcription, we've made cDNA from the microRNA, and with real-time qPCR, we've amplified the cDNA. These 2 reactions are sequence-specific, so we only amplify the cDNA of the microRNAs that we want to measure. In the qPCR, with each cycle, the copy number of cDNA duplicates. And the reaction produces fluorescence, which we can detect with the camera. The CT value is the cycle where the fluorescence, which correlates with the number of the cDNA, reaches the threshold. The fewer cycles it takes, the more cDNA we had in our starting sample. Using these curves, we can compare different samples' microRNA expression to each other. We've calculated the delta CT value by subtracting the geometrical mean of the 2 control CT from the CT of the target microRNA, and then statistically analyze the results with Ampere t-test. To understand the results, we need to discuss what the p-value is. It is the probability of getting these results just by chance. And we call the results significant if the p-value is less than 0.05. Based on these standards, we've found a significant difference in expression in the case of miR 130B3P and 194-5P. However, in the case of miR 96-5P and 106B, we found no significance. In the future, we plan to collect more samples, especially G3 ones, and measure 4 other microRNAs expression in tissue samples. In the long run, we also plan to measure the circulating counterparts of the significant microRNAs in blood samples. I am grateful to my mentors and coworker for their contribution to my work. Finally, I would like to show you some pictures from our lab. And it seems a very big building, and actually it used to be the biggest in Hungary, but compared to the world's other tallest buildings, it is still very small. Thank you for your attention.